Okay, let's just try and recall exactly what enzymes are. So this little Pac-Man guy here, we'll imagine that he's an enzyme. Now enzymes are proteins, which means they're made of long chains of amino acids. They coil up into this sort of structure where they have some specific areas. So the first area we're going to talk about is this area which is his mouth, which is going to be known as the active site. So I'm just going to write AS for active site. So he's the enzyme and there's the active site. Now enzymes, they catalyze reactions without being changed themselves. So if we have a substrate right here, what will happen is the substrate, which we'll label as S, will move into the active site of the enzyme. The enzyme will cause a reaction to happen, in this case splitting the substrate into two separate products, which then come out from the enzyme active site. So nothing gets changed, the enzyme can then go on and do another reaction straight away. So this is the product here. I think that's pretty much it. We saw this in the lesson where we had enzymes which were in the form of inside the living cells of the yeast. So we had the yeast in the bottom here, and then we added in some substrate, which was the H2O2. That gets added in to the yeast. The yeast enzymes reacted with the H2O2 to split it into H2O, which is water, and oxygen, which is a gas. Because we added in some fairy liquid, you'll remember that you had a lot of foam coming out the top, and it looked a little bit like elephant's toothpaste. Now we've seen the reactions happen, we can sort of start to interpret the results of different experiments. So if we're measuring the height of the foam of various different experiments, so you can imagine that we're adding hydrogen peroxide into various different concentrations of enzymes, we will notice that the height of the foam will increase the more enzyme is there. So the height of the foam is the dependent thing that we're measuring, so it always goes on the y-axis, and the concentration of the enzyme is the thing that we're changing, so that goes on the x-axis. As you can see, as the concentration increases, so does the height of the foam. The more enzyme there is, the faster the reaction, the bigger the bubbles, more foam there is. Now if you think about the second one, now this time the amount of enzyme is fixed, but we're changing how much substrate we're putting in. So we're adding in more hydrogen peroxide and then seeing if it makes any more foam. Now you notice as the concentration increases initially up until about 50% concentration of substrate, that the height of the foam was increasing, but then it starts to level off here. Okay, now enzymes, they're obviously like only got one active site per enzyme, and the problem is that that enzyme or active site can only do so much work. So even though we keep adding more and more substrate in, it can't break the substrate down any faster than it already is. So therefore it's doing as much work as it can, so the highest the foam will rise to is 10 centimeters, that's as fast as the enzyme can work, and that's why it levels off. There are other things that affect the reactivity of enzymes. So if we draw another graph now, so this time here we'll have the height of the foam on the y-axis, so foam in centimeters, and then along the bottom now we're going to have the temperature of the enzyme, the temperature that the reaction takes place in. So you imagine that we've got them in lots of different water baths, and we've got various different temperatures. So here would be 0, here would be 100 degrees centigrade. Okay. Right. So as the enzyme starts reacting, then initially, because it's very cold, there's not much of a reaction going on, so the height of the foam is quite low. So we represent the graph being quite low. But as it warms up, then the molecules start vibrating and there's more collisions between them, and therefore the reaction rate increases. Okay. Now, because it's a biological molecule, it will keep increasing until it gets to its optimum temperature. Once it gets to its optimum temperature, it means that it's getting too hot, and now actually the molecule is vibrating so much that that chain of amino acids, which the enzyme is made up of, starts to break apart and it starts to change shape, and that's known as denaturing. Now denaturing stops any reactions happening, so as it gets too hot we notice the reactions gets less and less and less and less until it drops down to nothing. So that's why it drops down. This is known as the optimum temperature. Oh, I'm not going to have enough space, but optimum temperature. And by the time it's got to here, it's completely denatured. Whoa. On the last page of my PowerPoint, I've set you a problem where there's been a problem in the lab and I've mixed up which enzymes came from which organisms. So some have come from animals, which are fish, some have come from bacteria, which are obviously bacteria, and some have come from Plasmodium, which is a type of Protoctis, which is actually the malaria disease. Now, each of these enzymes has a different optimum temperature depending on what it lives in. So, if you were something that lived in, if you ever wondered about why we are at, our body temperature is 37.5 degrees, if I put that as 37.5 degrees C, and this one here would be enzyme activity, so enzyme. Then what we would notice is that the activity would increase as it got warmer. It would peak at 37.5 and then it would decrease like that. So the optimum temperature for human enzymes to work at is 37.5 degrees. What you've got to do in the problem that I've set you is use your knowledge to work out which of the different enzymes, A, B or C, came from the fish, which one came from the bacteria, and which one came from the Plasmodium. And I'll be really, really impressed if you could write a conclusion to how you worked out which one's which. Maybe you'd use the Google maybe you'd Google some of the different environments they live in, and let me know how you got on. If you get any problems with any of this, then please just send me a Moodle message. If the video is helpful, then let me know, and maybe I'll make some more. If it wasn't helpful, also let me know, and I won't waste any more time making them. Alright, I'll see you on Wednesday.